So thank you everyone for joining this journey to discover foodborne pathogens. I am Amandine Noer. I have done my PhD in bacterial zoonosis unit in France. Then I moved to China to work on food safety. And now I'm working in Gdańsk University of Biotechnology. So during 1 hour, I will share with you some of my knowledge about few of the most infamous pathogens, their history, and how they are transmitted. First technical issue. Yeah, it's okay. So first, I'm going to explain the concept of food safety as it was defined by the WHO. WHO stands for World Health Organization. And the definition of food safety is the access to sufficient amount of safe and nutritious food to sustain life and promote good health. So it stands on three major pillars that are all closely related to each other: the food safety, the nutrition, and the food security. Today, I will focus only on food safety. So for everyone to understand, I think it's essential to give some basic definition. First, what is a disease? So a disease is a disorder of a structure or a function, and it's the symptoms of the ingestion of pathogens. The pathogens are the agent responsible of the disease. And the foodborne disease, or FBD, it's when food and drinks are contaminated by bacteria, viruses, parasites, chemicals, or toxins. It is very important to understand better food safety because it can influence national economy, regional development, and security of humans, of course. In 2020, the WHO, or WHO, World Health Organization, have recorded that more than 200 diseases are linked or caused by unsafe food consumption. And the impact of this food consumption can go from diarrhea to cancers. So it's a very important topic. The quantification of food impact on human health is very essential to better understand its impact on everyday life. And a foodborne disease outbreak is defined as when two or more people are developing similar illness after the same food and drink ingestion. If the disease is very rare or very severe, only one case is considered as an outbreak. So epidemiology is an essential field to study the impact of disease. It is known that 1 in 10 people fall ill because of food every year. In this, so food safety, as you can imagine, has a huge impact on our life. And 420,000 people die every year because of food consumption. Around 1/3 of these deaths appear in the patient under 5 years old. Food and water consumptions also cause an estimated 33 million healthy life years lost. Healthy life years it's also called DILY, and I will explain a little bit later what it stands for. So epidemiology is essential for defining public health priority, ranking the disease, and inform the public. Because you have to rank the disease to know where you're going to spend the funding for research and for curing the people. So DILY is very essential in epidemiology, and it stands for Disability Adjusted Life Year, and it allows to calculate how much a disease will impact on your life. It is calculated by the addition of YLL and YLD, and I'm going to explain what are YLL and YLD. So YLL, or year of life lost, is the total of all the fatal cases due to one specific disease multiplied by the expected individual lifespan. And YLD is is the severity weight factor. YLD means year of life disability. So it goes from 0 to 1, and 1 means death. To summarize, DILY is the impact on a disease in a population, and it's calculated by the fatality rate plus the severity of the disease. Now that I clarify some of the basic epidemiological definition, let's move on how foodborne disease or pathogens are transmitted. So at first, the pathogens responsible for foodborne disease have different reservoirs. They can be found in environment, in food and feed plants, in food animals, and in pets and wildlife. The human can be exposed at different levels. So water and soil, foodborne, direct animal contact, and also human-to-human reservoir. I would like to stop here for a moment just to give you an important definition, even though it's not fully related to the topics today, but I think it's important for you to know about zoonotic disease. So what is a zoonotic disease? It's due to zoonotic pathogens, and it's a pathogen that can cross the species barrier. So it can go from animal to human and human to animals. And here you have few examples of famous zoonoses. So for example, I'm sure you heard about anthrax, salmonella, tuberculosis, also Escherichia coli that I'm going to talk about today, or Ebola and SARS. So these are just few examples. So about the foodborne disease, today I will mostly focus on the pathogens that are transmitted by food and by water. So foodborne pathogens are mostly responsible of diarrhea. It's the most common form of illness resulted of contaminated food ingestion. Every year, 550 million people fall ill because of ingestion of contaminated food, and 230,000 die. So today I will focus on three major categories of pathogens responsible of foodborne illness, so the disease, death, and DILY that I already defined later on. So I'm going to show you a table now with foodborne illness, foodborne death, and foodborne disability adjusted life year, or DILY. And here you can see in this table that are listed the most infamous foodborne pathogens in descending orders. So they are ranked by the number of cases, estimated number of cases. So you have bacteria in blue, viruses in green, and parasites in yellow, and I will try to define most of them today. So first, let's go to the first categories, the bacteria. So what are bacteria? Bacteria are microscopic single-cell organisms, and they have different structures, morphology, and belongs to different species. They are prokaryotic in opposition to eukaryotic. So what are prokaryotic and eukaryotic? Eukaryotic cells means that they have membrane-bound nucleus and organelles, when prokaryotic cells, like bacteria, have free-floating DNA. The bacteria also have usually two protective layers at the surface, and sometimes they have a protective capsule. It's like an extra layer that allows them to survive better. Sorry. So there is a very famous technique to distinguish different groups of bacteria, gram-positive and gram-negative. And this technique is called gram staining. It uses different stain, and the bacteria appears in two different colors under the microscope. So the gram-positive bacteria appears violet, purple, and the gram-negative appear pink. And this happens because the composition of the bacterial membrane is different. So the gram-positive bacteria have a cell membrane covered by a thick layer of peptidoglycan. Peptidoglycan is a large polymer made up of sugar and amino acid. When the gram-negative bacteria are covered by a cell membrane, recovered by a thin layer of peptidoglycan, and then at the surface they have an outer membrane which contains polysaccharides. So the structure of the membrane of the bacteria is visible under the microscope by this specific staining. They also have different interaction with their host and their environment that helps to classify them. So they can be symbiotic. It means they are useful for their environment and their host. They can be commensal. So they share the same space and resources within their environment and host. Or they can be pathogenic. So they are harmful for their environment and their host. So the first bacterium I will present today is Campylobacter. So Campylobacter was discovered by Theodor Escherich, a very famous German-Austrian pediatrician, in 1886. It was then isolated in Belgium and identified 1 year later as a human pathogen. It's one of the 4th global causes of diarrheal disease, and it's the most common bacterial cause of human gastroenteritis. The at-risk groups are the very young children, the elderly, and the immunosuppressed. So Campylobacter is responsible of campylobacteriosis, and you can see on this beautiful picture, like it's a spiral shape or S shape or curve or road shaped bacteria. There are 17 species and 8 subspecies referenced today. In this, the campylobacteriosis are principally due to Campylobacter jejuni and Campylobacter coli. And Campylobacter jejuni grow the best at 37 to 42 degrees. So it is perfectly adapted for human body. We actually have some issue with Campylobacters because there is a spread of antibiotic multi-resistant strains in human population. And this causes a lot of damage in human health. So the principal source of contamination to get Campylobacter is the ingestion of poultry, raw meat, and just know that Campylobacter is a zoonosis. So how does Campylobacter symptoms look like? So generally, they appear 2 to 5 days after ingestion, and they cause diarrhea, fever, abdominal cramping for 3 to 6 days. In some rare cases, it can be associated with Guillain-Barré syndrome. Guillain-Barré syndrome is an acute neuromuscular paralysis, and the rate of infection increases with the ingested dose. So more you ingest bacteria, better are your chance to have more symptoms. So what is the cycle of Campylobacter? So you ingest the bacteria and then it goes to your intestine. In the intestine, Campylobacter pass the mucous barrier, and then if there is a disruption of the epithelial barrier, it can go through to reach the submucosa. If not, it can attach to the cells, and then it is internalized and reach the submucosa. There is cytokine production. These are small molecules that can attract your immune cells. So your immune system is activated. Then the bacteria can go inside the cells of the immune system, and there is an inflammation that causes diarrhea and then causes the clearance of the bacteria. So a few fun facts about Campylobacters. As few as 500 bacteria can make you ill, so it's a very low number. The disease had different names during the history, so it was known as cholera infantum. Why cholera infantum? Because the epidemiology of the disease was linked to the end of breastfeeding. So when babies started to eat different kind of food, they get healed. They had cholera, so like diarrhea. So it was called cholera infantum. It was also called summer diarrhea, and is referred as vibrio-like organism due to its shape and due to its shape. So the second bacteria I will present today is the very famous Escherichia coli. I'm pretty sure most of you have heard about it. So Escherichia coli was also discovered by Theodor Escherich in the feces of neonates in 1885. The at-risk group are the young children and the old people. Old is over 65 years old, so it's not that old. It can cause mild to severe gastrointestinal illness, and it's dose and type dependent. We have different types of Escherichia coli. I will briefly show you a few of them. So they are due to, so usually Escherichia coli is living in the animals and human intestine, and it's harmless. There are more than 700 serotypes identified of Escherichia coli, and it's a gram-negative rod shape, as you can see here on this electronic microscopy here. And Escherichia coli can produce secret toxins. It can also grow with or without oxygen. Some very famous pathogenic Escherichia coli are STEC for Shiga toxin producing Escherichia coli, or ECEH for enterohemorrhagic Escherichia coli. The principal source of contamination are the ingestion of contaminated aliment with feces. So it can be raw or undercooked meat, for example, dairy product or untreated milk, fruits and vegetables. And there is also a human-to-human transmission due to the lack of hygiene. So the symptoms appear usually 3 to 8 days after infection. And it causes severe stomach cramp, diarrhea, fever, nausea, and vomiting. And as I told you before, the severity of the symptoms depend on the type of Escherichia coli you ingested. So here you have different kind of pathogenic Escherichia coli presented here. So enteropathogenic, Shiga toxin producing Escherichia coli, enteroaggregative, diffusely adherent, enteroinvasive, adherent invasive, and enterotoxigenic. What is interesting is that they all have a different way of invading your body and causing the disease. But in every case, they first have to adhere to your cells. Then they're going to produce toxin or create lesion depending on the type they belong to. And these toxins production or the lesion will link to water and electrolyte release, and this will cause the diarrhea. So a few fun facts about Escherichia coli. There are 11 prestigious Nobel Prizes associated with Escherichia coli. So you have the list here. I won't go through the details. The only important things we need to remember is that why so many Nobel Prizes? Because Escherichia coli is a very nice model in the lab. It grows very well, and you can play with that bacteria. So it's an amazing tool for research. The most notorious type of Escherichia coli is O157H7. So it comes from bovine, and it's responsible of food intoxication that can be potentially mortal. And I'm sure that you already heard about this bacteria, like this E. coli type, on the news, because it's very famous. Escherichia coli was also used to make useful substance, like human insulin. So insulin is a hormone that controls the amount of glucose in your body. Human growth factor, which is a factor that stimulates the proliferation, the healing, and the cellular differentiation. Taxol as well, and Taxol is a cancer medication. And epidermal growth factor, which stimulates the growth of the tissue. So now let's move on to Salmonella. So Salmonella is the typhoid fever living agent that was suspected by Karl Liebermeister in 1875. In 1879, the bacilli of Salmonella was discovered by Carl Joseph Ebert in lymph node and spleen, and it was named after Daniel Elmer Salmon, which was a veterinary pathologist and administrator of research group in USA. Salmonella is the most common source of foodborne outbreaks in European Union, and 67% of this outbreak appear in Slovakia, Spain, and Poland. So again, the extreme age group are the most at risk. And Salmonella can be responsible of salmonellosis when it's due to Salmonella typhimurium and Salmonella enteritidis, or to typhoid fever when it's due to Salmonella typhi. It's a gram-negative, rod-shaped, non-motile bacteria. Non-motile means it's not able to move. So the principal source of contamination are the lack of hygiene. When you're touching contaminated surface or you're not washing your hand properly. The raw and the undercooked food, especially eggs, chicken, beef, fruit and pork and vegetables. And it's very common when you have warm weather and you do not put your food in the refrigerator. There's also animal to human contact transmission because dogs and cats can carry the bacteria. So do not kiss your dogs and cats. This will be my advice. So salmonellosis symptoms appears after 6 hours to 6 days after ingestion, and they usually last for 4 to 7 days. They provoke gastrointestinal illness with diarrhea, fever, and stomach cramp. So how does the cycles go? So you ingest the bacteria, it goes to your intestine, then it crosses the mucous barrier, and it adheres at the surface of the cells by some molecule called adesin. The bacteria then goes inside the cells by a different way. It is encapsulated in small vesicles inside the cells, and it can multiply inside these vesicles, and then it is released in the submucosa, and then it's taken by phagocytes. Phagocytes are part of your immune system, and then it moves to the bloodstream. There is a very famous, infamous story about salmonellosis. Is the infamous story of Marie Malone. Marie Malone lived between 1869 and 1938, and she was a cooker in USA. What happened is that Marie Malone employers were falling ill, and they had fever and diarrhea, and few of them even die with typhoid fever. So the health agency have seen a link between these people falling ill of typhoid fever and the fact that Marie Malone was cooking for them. And they've done some research, and they find out that she was carrying Salmonella typhi, but she has no symptoms. So she was the first healthy carrier identified. And a healthy carrier is defined as healthy living being carrying a pathogen without any symptoms. After this, she was forbidden to cook, but she was in the total denial because she wasn't ill, so she didn't think she was carrying the bacteria. So she get back to her old job, and then again people were falling sick. So she was forced to quarantine. And then after a few years, she was released with the promise to never cook again. But she never kept the promise, and she cooked again, and a few people fall ill again. So she was interned for the last 2 decades of her life. What we know about this case now is that at least 3 people died because of her. 51% were infected, and she spent nearly 30 years of her life in isolation. So it's kind of a very sad story. So now let's move to Shigella. So Shigella was discovered during a seasonal dysentery in Japan by Kiyoshi Shiga. During this episode of dysentery, more than 90,000 cases were reported, and the mortality rate was over 20%. So the dysentery is very common in a tropical region, and it's endemic in developing countries. It's possible to have seasonal epidemic. The mortality is higher when people are older than 5 years old. So Shigella causes shigellosis, and shigellosis are due to 4 species: dysenteria, flexneri, boydii, and sonnei. It's a gram-negative, facultative anaerobic, non-spore-forming rod with virulence plasmid. So a facultative anaerobic means it can grow without oxygen. It's possible. Non-spore forming, it means, so the spore is a resistant form of the bacteria. So it is not able to form spores. And rod is just the shape, as you've seen before. So the principal source of contamination is the human reservoir. So when you have a lack of hygiene, you can be contaminated by fecal-oral route or by ingestion of contaminated food and water. And Shigella is extremely infectious, as only 10 to 100 bacteria are able to infect a human, adult human. So what are the symptoms of dysentery? It's abdominal pain with very heavy diarrhea, as you can go up to 100 times in 24 hours to the toilets. And sometimes this diarrhea is even hemorrhagic. So in some very rare cases, when you have ingestion of Shigella flexneri, you can have Reuter's syndrome, and this is a reactive arthritis. So usually the symptoms appear 12 to 96 hours after ingestion, and they last for 5 to 7 days. The infection remains 1 to 2 weeks after the symptoms disappear. So what is the cycle? So as I showed you before, like always, the bacteria have to enter one of the cells, multiply inside the cells, then cause cell death. When it causes the cell death, it activates pro-inflammatory signal, and then you activate the immune system, and the bacteria in the meantime is able to go back inside the cells and to cross 2 different cells and continue the cycle. I won't go through many details because I don't have much time, but I will leave you the references so you can check later on if you are interested in that. So a few fun facts about Shigella. It's genetically very closely related to Escherichia coli that I presented to you before. The term dysentery, which means bloody and mucus-containing stool, was introduced by Hippocrates. And a common ancient thought was that this disease was only of tropics and war. In fact, Shigella, and dysentery in general, has an important role in military operation as well as in jail, prison, and asylum. It is known that it kills or incapacitates more soldiers than the battles. And there is even a code of honor during the civil war in USA that was like soldiers were not allowed to shoot other soldiers when they were pooping. So the reason for that is that it's a code of honor, so it's not very fair to shoot someone which is pooping. But the other reason is that if someone was having dysentery, the chance that he was going to die soon was very high. So why losing the bullet for someone which was already almost dead? So the last one I want to talk about, the last bacterium I want to talk about is Vibrio. So Vibrio was discovered during an outbreak in Florence in 1854 by Filippo Pacini. And Vibrio cholera, so probably the most famous one in this species, was discovered by Robert Koch in India in the Ganges water. The at-risk group are the immunodeficient people, and Vibrio is responsible of vibriosis. And vibriosis is due to 3 significant species. When the disease is cholera, it's due to Vibrio cholerae. But when you have acute enteritis, so non-cholerae, the most common non-cholerae Vibrio is Vibrio parahemolyticus. And there is another one which is less common but more lethal, named Vibrio vilnificus. So Vibrio is a gram-negative, facultative anaerobe, highly motile, with 1 flagell in one hand. So this bacteria is able to move. And usually it has shape or spiral, it has S shape or spiral shape. Principal source of contamination of Vibrio is the water, because Vibrio is an aquatic organism, and you can get it by ingestion of raw or undercooked seafood, or when you expose a wound to seawater. So what are the symptoms of Vibrio? You have watery diarrhea, abdominal cramping, nausea, vomiting, and chill. And they appear 24 hours after ingestion. Usually they last for 3 days. In very rare case, you can have severe illness. It's possible to have skin infection when you go to the water with an open wound and the water is contaminated by Vibrio. So how does Vibrio invade your body? So you ingest it, it goes to the small intestine, then it passes the mucous barrier, attached to the cells and lost its flagella in the meantime. When it's attached, it's able to produce some toxin. The toxins are going inside the cells and they force the cells to release chloride ions. This release of chloride ions make unbalanced osmosis, and this creates diarrhea. And then the bacteria detach and go to the next target. So a few fun facts about Vibrio. They are halophilic. It means they love salt. And they are mostly found in temperature when the temperature of the water is rising, because they attach to algae. So here you can see in black the water temperature, and in white the quantity of Vibrio in the water. And as you can see, the level of Vibrio in the water can be predicted like the weather, because it's linked to the climate change. So the next category I want to talk about of pathogens are the viruses. They are small genetic codes surrounded by protein coat, like microscopic parasites. Then the nature of their genetic information encodement is very important, as they give us information about their behavior, their classification, and their nature. So the genetic information is encoded in 2 different forms, like major forms, DNA and RNA. I won't go through the details because I won't have time, but I just want to give you some basic definition. So DNA stands for deoxyribonucleic acid, and it's 2 twisted strands of genetic information in a spiral shape called helix, as you can see in group 1, one example of what it looks like. And RNA for ribonucleic acid is similar to DNA, but it's single strand. So as you can see in group 4. I won't go through details, just know that all the viruses have different way of coding their, all viruses have different genetic information or encodement. And this genetic information will give information about the proteins that has to be produced to make a new virus. So as viruses are only genetic information, they are non-autonomous. So they need to invade a cell to use its replication machinery to replicate. And they cannot survive outside of an host. Due to their small size, only a few nanometers, and even if their existence was suspected for a very long time, they had to wait for major optical technology progress to prove their existence. So the first visualization date from 1931 with the electron microscope. And finally, viruses have different structures which allow to classify. So when you associate the genetic information encodement to the structure, then you have a classification of viruses. But it's very tricky, so I won't go through details. I just want to show you the diversity of the viruses, the different structures that they can have. So you have here a few examples. I'm going to present only 2 viruses today. So the first one is hepatitis A, or HAV. The first suspicion of this infectious agent date of 1908, sorry, and it's the first isolation date of 1973, and its characterization in 1978. So the at-risk group are international travelers, homeless people, HIV carrier, and drug user. HIV stands for human immunodeficient virus. Interesting fact is that when you are under 6 years old, only 30% of the patients are showing symptoms. But when you're older or adults, 70% are showing symptoms, and the most common symptoms of hepatitis A is jaundice. I'm going to explain later on what is jaundice. So this virus is endemic in Central America and South America, Africa, Middle East, Asia, and Western Pacific. The principal source of contamination is the human itself, because human is the only natural host of this virus. So you get it by fecal oral transmission and with liver replication. So just few information. Hepatitis A is a RNA positive piconovirus. And it's, when you look under the electronic microscope, you can see a small spherical particle of 27 to 32 nanometers of diameter. This virus is stable in environment for months, and especially when the pH is low and the temperature is moderate to frozen. So what happens when you've got when you ingest hepatitis A? So you have an incubation period of 28 days on average. Then you can find the virus in the feces. And after 10 to 12 days, you can find the virus in the blood. Usually, it does not last longer than 2 months. In some cases, you can find the virus up to 6 months after the resolution of the symptoms. So what are the symptoms of hepatitis A? It's an abrupt onset of fever, malaise, anorexia, nausea, abdominal discomfort, dark urine, and jaundice. So jaundice means you become yellow. And jaundice was first described by Hippocrates. Jaundice comes from jaundice, which stands for yellow. It also can be called icterus, and comes from this bird, golden thrush. So one of the ancient Jewish and Greek belief is that when you got jaundice, you need to catch this bird and place it nearby your umbilicus to resolve your condition. And this is due to a simple fact. You are yellow and the bird is yellow, so it was believed that yellow will attract yellow and the yellowishness will get out of your body if you have the bird on your umbilicus. But it was just a belief. So this jaundice triggers attention to medical practitioners for over 5,000 years. So the next virus I want to talk about is norovirus. The first outbreak of this virus was identified in Norway in 1968, and the virus was identified in 1972. So the extreme age are at risk group, as I showed you before, so under 7 years old or over 65 years old, and it's human enteric pathogens. So you are contaminated by fecal oral route, vomit, contaminated water, and food ingestion. So here's the map of the death due to norovirus by countries. The norovirus is an RNA-positive Calliciviridae virus, so this is just its classification. It's small. It shows small virus particles of 27 nanometers, and it's very stable in the environment. So when you ingest norovirus, you have incubation period of 48 hours on average, and usually the illness lasts for 4 days, and the resolution appears 1 to 3 days after. If you have comorbidity, then it can be a little bit longer. The virus is excreted in the stools for approximately 4 weeks to 1 month after the resolution of the symptoms. And of course, when you have symptoms, you have more viral particles in your body, so you find more particles in your stool. So the symptoms of norovirus infection are watery diarrhea, vomiting, abdominal pain, and nausea and fever. So few fun facts about norovirus is that only 100 viral particles are sufficient to make adult human sick. So very small inoculum. And in 1929, it was describing as a winter vomiting disease. And there is a very interesting project that was done by Catherine Mackinson-Booth in UK about the spreading of norovirus. So norovirus, this researcher, she wanted to understand how far you can spread norovirus when you're vomiting. So to do so, she created vomiting larvae, this small body here, and this body is able to reproduce the way you vomit when you get norovirus. And she measured how far the vomit was spreading, and she realized that vomit can fly more than 3 meters away from the mouth. So if you see someone vomiting, don't get close to them. So the last category I want to present is the parasites. So parasites are multi or unicellular organisms. They can be few centimeters long, and they live in or on a host organism. There are 2 main classes that can cause foodborne disease. The protozoa. So protozoa are microscopic one-cell organisms like Giardia here. This I'm going to describe it just right after. And helminths, so they are large multicellular organisms, and they can be like flatworms, thorny headworms, and roundworms. And there is like a nice example of helminths here is schistosoma, but I won't describe it today, even though the picture is amazing. So there are a lot of different species of parasites, and I won't have time to explain all of them, so I will just describe 3 of them today. So the first one is Giardia, here, beautiful. So it was described for the first time by van Leeuwenhoek in 1681. And this researcher was very dedicated to his job because he described it by digging his own stool. So he took his poop and he analyzed his poop and he found Giardia inside. The at-risk groups are the close contact people, the people working in childcare, the travelers, the campers, the swimmers, and the one in contact with animals and environment. The prevalence of Giardia varies a lot depending in which country you are. So it's quite low, like 2 to 3% when you are in industrialized country, but it can be very high when you go to low-income or developing country, as it can be up to 30% prevalence. So Giardia causes giardiasis, and it's due to two major protozoa. So Giardia enterica, or Giardia lambia, that can also be called Giardia intestinalis and Giardia duodenalis. So these are the same, different name for the same organism. So it's a worldwide protozoan, and usually it rises during the late summer in USA. As you can see here, so it's flagellated, it has a nice spear shape, and it got two active nucleus here and here, like two eyes. So Giardia is a very common cause of waterborne diarrheal disease, and it easily spread by mouth contact with contaminated water, surface of people, or people. So do not kiss people in late summer in USA. The most common intestinal parasite is the most common intestinal parasitic disease. More than 1 million people per year catch Giardia. And there are some possible zoonotic transmission. So interesting thing about the parasite is that usually they have a complex life cycle. So how does it work? So Giardia has two major forms, the invasive forms and the infective forms. So the invasive form are the cysts and the invasive form are the trophozoite. So you ingest cyst, which is the survival form in the environment. And then you have an incubation period of 1 to 2 weeks, and then diarrheal disease started. During this time, the cyst is going to small intestine to exist. So it means from the cyst form, it becomes a trophozoite. So the trophozoite is the invasive form. Trophozoite is moving to intestinal lumen, where it maturates and multiply. And then goes to the colon, where it encysts again, so it becomes again a cyst. And then it's excreted by stool. Sometimes the encystation is not well performed, so you still have trophozoites that are excreted in the stools. If trophozoites are going outside your body, they just die because they cannot survive in the environment. But when you excrete the cyst, it goes to the environment and it can survive from weeks to months outside the host. And when it survives, then it is able to be ingested again by a new host, and so on and so on. So a few fun facts about Giardia. The first detailed description was published by Lamb in 1859, and it's why we call it Giardia lambia. The ancestor of Giardia was very complex organism, and then the evolution make it become less complex, so more simple. And also Giardia can eat or expel through organelles. So it has no mouth, but it can take things from outside his body and release things in the outside environment. The second parasite I want to talk about is entamoeba. Entamoeba was discovered in 1859 by Lamb and proved to be pathological in 1875 by Lorch, who was studying young Russian farmers suffering of chronic dysentery. There are many different species of entamoeba, and they all can infect different kind of hosts. I won't go through details. I just write the list down for you to have an idea of the diversity of entamoeba. So the prevalence is higher in temperate, subtropical, and tropical climates. And the at-risk groups are the immigrants or the travelers from tropical countries or the people living in poor sanitary conditions. So every year, 50 million cases worldwide are referenced, and 55,000 to 100,000 people die every year of entamoeba infection. So approximately 10% of the global population will face infection with entamoeba during its life. It's transmitted by a fecal-oral route or via ingestion of contaminated food and water. So entamoeba causes amoebiasis. Entamoeba is a protozoa with two stages. The motile trophozoite, here you can see, is the invasive form of the disease. And the second stage is the cyst, which is the infective form. Entamoeba histolytica is the most pathogenic entamoeba, and it's responsible of amoebic dysentery or amoebic liver abscess. So two different forms of the disease. Entamoeba can be discriminated under the microscope by counting the number of nuclei, because entamoeba can have 1, 4, or 8 nuclei. So it's a very important tool to differentiate the different species of entamoeba. So what are amoebiasis symptoms? They usually develop 2 to 4 weeks after the ingestion, but only 10 to 20% of the people that ingest entamoeba will become sick. The symptoms are abdominal pain, fever, hepatomegaly, diarrhea, ulcer, and abscess. So how does it work? So you ingest the cyst form here. It goes to your stomach, then to your intestine, where it's able to exist, so become a trophozoite stage. This trophozoite stage can multiply and then migrate in the big intestine. And then two things can happen. Usually, you excrete the trophozoite via the stools, or you excrete the cyst via the stool. If you excrete trophozoite, you're going to have diarrhea. If you excrete cyst, you're going to have formed stools. And then the trophozoite is the dead end, so the parasite cannot survive in the environment. But if you have cyst, then it goes back to the cycle again and again. But there is another option. So the trophozoite form can migrate in extra-intestinal organs and create extra-intestinal disease. Most of the time, it creates abscess, for example, in the liver. Here you have a nice example. It can also reach the brain or the lungs. But these forms are rare compared to the intestinal disease. So fun fact about entamoeba, only 1 to 100 cysts are recurred for amoebic dysentery in animals model. So it's quite low number of parasites to get the animals sick. And something interesting as well is that trophozoite is able to eat your red blood cells. So the last one before the lunch is Ascari. So Ascari was described for the first time in 1758 by Carl Linnaeus. Carl Linnaeus is a Swedish scientific known as the father of the modern taxonomy. And taxonomy means classification of organisms. The principal source of contamination are the humans, dogs, and pork. And it's worldwide distributed, as you can see here. So it's the prevalence of Ascaris lubricoides infection here. So lighter the color here is, lower the prevalence is. And it's endemic in South America, Central America, Africa, and Asia. It's often in tropical and subtropical region, as you can see on the map. So 870 million people to 1.2 billion people are infected with Ascaris. It's contaminated by fecal oral route, by ingestion of contaminated products, mostly water, food, and agricultural products. And it's always associated with high human density and lack of hygiene. So Ascaris causes ascaridose, and it's principally due to two different species, lumbricoides in human and some in pork. So Ascaris is an helminth. It's a parasitic worm, as you can see here on this picture. So it's like a very nice worm. An interesting fact is that Ascaris is sexually oriented. So it got adults, male and female. And when they reproduce, the female are able to lay oval or round eggs. Here are a few examples, like 2 examples of what the eggs look like. So what are the ascaridose symptoms? They are non-specific abdominal pain, and they can be associated with intestinal obstruction, nausea, diarrhea, and abscess. Usually they are asymptomatic, but the severity is always associated with the parasitic charge. So the symptoms appear 6 to 8 weeks after the ingestion and lasts for about 3 weeks. You have to get infected by Ascaris to swallow infective stage. It goes to the gastrointestinal tract where it maturates and it becomes all male adults or female adults. From there, usually you excrete the eggs by the stool. If the eggs are unfertilized, then it's a dead end and nothing happened. But if the eggs are fertilized, then they go through maturation stage in the environment before becoming infective stage and being able to contaminate human again. In few cases, you can have migration of larvae in the body. And I just want to show you a nice example here of the migration of the larvae in the eyes. But this kind of migration is an accident because it's a dead end for the parasite. It cannot grow there. So a few fun facts about Ascaris is that one female can release 200,000 eggs per year, fertilized or not. So it's a lot of eggs. The fresh stools does not contain infectious eggs. As I told you, the eggs, when they are released, they need to maturate for 2 weeks to be able to infect humans. In dry and windy weather, the eggs are suspended in the air and can be swallowed. In the environment, the eggs are viable for 6 years in temperate climates and 40 days in extreme conditions. So I'm reaching the end of the topic. I hope you learned a few things. I just want to give you like a prevention message. So just think global because diseases are spreading everywhere, but you can act local to prevent the disease. Finally, here is some advice for having safe food. So always keep the water safe, keep the food at safe temperature, cook food thoroughly, and finally separate raw and cooked food. And of course, don't forget to wash your hands. So I hope this talk opened your appetite for the rest of the day. If you want, I leave the reference that I use for the talk here. So I don't know if this presentation will be shared or not, but you can have a look. And I thank you very much.
Thank you.